various, all their data. And that's what's schematically shown by these arrows here. And there are at least a few different activities that go into developing these resources, right? One is clearly software development. Second one is sort of the actually standing up these instances in terms of the system administration and so forth. And the third piece is the annotation efforts that are very specific to each community. And as Scott alluded to, right, what people undoubtedly realized was that on the software development side of things, there's a lot of synergies to be had between all these different communities. And so hence GMOD and all the tools and so on and so forth. And of course, based off of that history, as Scott's alluded to, right, GMOD has been fantastically successful. I just went to this GMOD users page on the wiki, and there are 199 organizations listed as on that page. So clearly, fantastic success. And the question that the provocative question I want to pose is, does this current model scale, right? With all the genome sequencing that's currently being done, right, what are the bottlenecks that we're going to face in the future? And just to put this in a very real way, here I'm showing 96 genomes. This is more in line with the number of genomes that are currently sequenced. So we have about 3,000 genomes currently sequenced. And just to look at the history and project forward a little bit, we are currently here, and again, the sum total of bacteria, eukaryotes, or archaea, are about 3,000. Just as milestones moving forward, right, we're going to hit 10,000 sequenced genomes in roughly 2015, 100,000 sequenced genomes in 2022. If history holds, we double the number of sequenced genomes roughly every 2 years. So clearly, a lot of genome sequences being developed. And so, again, if you come back to this picture, right, okay, again, GMOD is a fantastic software infrastructure to build upon, but every one of these efforts also has some administration associated with it, right? Standing up the databases, putting in the web servers, installing JBrowse, gbrowse, whatever. Those are all things that each community is responsible for. And you might argue that these organisms here, totally fine, right? NIH has invested significant resources in each one of these communities, so there are dedicated bioinformaticians for these model organisms that can do this effort and at the same time contribute to GMOD. But what about all the organisms for which there will never be sort of large NIH-funded model organism communities? What happens there? Just to give a sort of a case-in-point example, I call this the long tail of genomic data, right? So in 2009, there was this prominent paper using affy-tiling arrays to look at the transcriptional landscape of Listeria. And very detailed work, highly cited, yada, yada, yada. And among all the findings that were reported, right, they identified 517 operons, 103 small regulatory RNAs. So important data that might be represented in a GMOD-like database. And you might think that it would be that those features would be in the Broad's Listeria database, or in NCBI Genome in their GFF file, or Ensemble Bacteria, or in the UCSC Microbial Genome Browser. But in fact, that data is not anywhere, right? That's not in any of those databases the results of this finding. Thankfully, right, this paper was published in a high-profile journal, Nature, and so we can get the data out in structured form. Until, of course, you realize that actually it's buried in a PDF and supplementary table. Right? So this data, right, is fantastically useful, of high use to the community, not only in Listeria, but related organisms when you want to do comparative genomics, and yet the data is buried, right? It's not accessible in any real way. So, again, to come back to this model, right, what happens when we start, right, and so the idea is, right, can we essentially starts to develop common infrastructure, which we provocatively, tentatively named centralized model organism database, that essentially allows all organisms to essentially feed into some common infrastructure. And those that not only could service these well-funded model organisms, but also the long tail of organisms that are being sequenced and will continue to be sequenced in the future. Of course, as Scott has introduced earlier today, right, there's lots of relevance to efforts like GMOD in the cloud and GMOD in the box, and we can talk about what those synergies and complementary approaches are later. But if you want to take the cloud analogy moving forward, right, I sort of think of this as GMOD as a service, right? If you like gas. So CMOD, gas, I'll let you guys choose, right? So this is really what the central premise that we wanted to explore in this idea. So I can't give an entire talk just hand waving. So at this point, I need to take a detour to actually tell you things that we've actually done in this space that are relevant towards crowdsourcing and organizing biological information. And then we'll circle back into how that relates to CMOD here. Okay, so my group is very interested in human gene function, and we did an analysis, we continually do this analysis, and this basic picture stays the same, essentially of gene ontology annotations as a proxy for structured data about genes. And if you sort all 27,000 genes in order of the number of linked Go annotations, so these are the most highly annotated genes over here, these are the long tail over here. At the top of this list, these are the most highly annotated genes, and it's sort of a who's who list of biomedically relevant genes. And these have hundreds of linked Go annotations. But what was interesting and notable to us is how fast this drops off, right? To the point of 65% of genes have 5 or fewer linked gene ontology annotations. 41% have 1 or 0 linked gene annotations. So it says in our effort to systematically describe the function of human gene function, right, we've got a long way to go, right? And there are many reasons why this pattern could exist. I'm going to suggest at least one of them is because the literature is sparsely curated, right? So this is a picture we're all familiar with. The number of PubMed index articles published per year is going up rapidly. I think in 2013, I haven't updated this in a while, in 2013 we passed over 1 million new PubMed index articles every year. In total, I believe we're up above 22 or 23 million PubMed indexed articles. In the face of this explosive growth of the biomedical literature, our ability as individuals to consume the literature hasn't really kept pace. We pretty much read the same number of articles every single year, right? So what does that lead to? Well, it leads to this bottleneck in our biocuration, right? Where clearly the NIH isn't going to increase the amount of biocuration resources exponentially, as we've learned. And so it becomes more and more difficult for biocuration to keep up. So as one metric of that, right, if you look at all Go annotations and look at the number of linked PubMed references to those Go annotations, you find 311,000 linked PubMed annotations. That's 1.5% of PubMed. If you believe that more than 1.5% of PubMed is relevant to gene annotation, then it says we're under-annotated in some way. So this is sort of the bottleneck that we've noticed. We aren't the only ones to notice this. This is certainly well appreciated within the biocuration community itself. In this review article about the future of biocuration, they note this problem and many other and many possible solutions. And one of the solutions they explicitly reference is quoted here. Say, sooner or later, the research community will need to be involved in the annotation effort to scale up with the rate of data generation. So this idea of directly engaging large communities, right, is not particularly new, and it references this concept of the long tail, which I've mentioned a couple times, but let me explain in a little bit more detail. Right, the long tail is actually best explained first by considering its opposite, which is the short head. Okay, so the short head is when you have a few number of contributors, each contributing a lot of content. Okay, and the classic short head type of institution, I would say, is newspapers. Right, there are about 1,000 daily newspapers still in the United States, and each of those produce an amount of content, a substantial amount of content every single day. The counterpoint to that is the long tail, where you have a large number of contributors, each of whom contribute a relatively small amount of content. But even though their contributions individually are small, collectively, the long tail can be quite large. So here we have the counterpoint being blogs. So there are very few blogs that produce as much content as a daily newspaper. But blogs in aggregate, right, there are over 100 million blogs online now, and in aggregate you can get a lot of content out of blogs. So this general pattern between short head and long tail is not limited to news reporting, right? You can see this in video generation between TV and Hollywood studios and YouTube. You can see it between consumer reports and Amazon reviews, food critics versus Yelp, so on and so forth, right? You can see all sorts of examples of the long tail in sort of modern technology. Undoubtedly, the most important and the most successful long tail application is Wikipedia. Okay, so Wikipedia, of course, is the online encyclopedia that anybody can edit. There are only two things I'll note in the purpose of this talk. First is that Wikipedia is reasonably accurate for science. Right? So Nature did this study several years ago, comparing Wikipedia to Britannica Online for a series of science-related topics, and their conclusion was that the scientific accuracy was roughly the same. But what you get out of a very open model is that you get the incredible breadth and depth out of Wikipedia, right? So these are old statistics, 2008 now, but even back then, right, both in terms of the number of articles and the number of words, right, Wikipedia far outpaces Britannica Online, a prototypical shorthead type of application. So the fundamental hypothesis that we wanted to approach was, can we use the long tail scientists to directly participate in this gene annotation process? So again, to come back to the problem statement, right? Lots of times, through high throughput studies, genome sequencing, whatever, we get to a list of genes that we are newly interested in, and very often the first thing is to plug that into PubMed. So to take an extreme example, if I am interested in fibronectin, I search PubMed, I get out 30,000 research articles, right? Pretty daunting to try to get a lay of the land of what's going on, what's relevant for fibronectin. Typically we rely on, some esteemed professor somewhere to write a review article to periodically summarize that literature. And our question is, can we use the community to essentially summarize that into a single Wikipedia article, right? That is the point of Wikipedia, to write an encyclopedic article about topics of interest. So, this is what we wanted to do. With any crowdsourcing application, I typically like to think about this positive feedback loop that I'm going to describe here. So, in order to get this process started, right, we called this the GeneWiki. We needed to create a GeneWiki page that had some level of utility, right? We need to provide some value to people. And if we provided some value, then over time, we'd attract some number of users. Out of those number of users, some number of those would actually stay around to make an edit. It could be a very small percentage, but some percentage will, if you enable them. And that could be as significant as summarizing a recent paper in literature. It can be as trivial as fixing a typo. But regardless, in the context of making that contribution, you make the GeneWiki page that much more useful, which will draw that many more users, more contributors, and so forth. So this is that positive feedback loop that we're trying to engender within our crowdsourcing effort. So along that first axis, how do we establish utility? Well, the first thing we did, and we initiated this effort in about 2007, 2008, was to take structured data that was already out there in structured databases from NCBI, from Ensemble, Unicron, and so forth, and essentially integrate that, reformat that, and then put that systematically into GeneWiki pages, right? So we can do this en masse, and we started with about 10,000 gene stubs. And we can do this essentially using highly automated methods. And the idea was that we thought that people would be much more likely to edit a page that already existed as opposed to creating a new page that didn't exist previously. So based off of that, how do we do in terms of usage now? Well, usage, we'd argue that because it's within Wikipedia itself, we piggyback on that. So in total, these get viewed about 4 million times per month. Clearly, there is a wide distribution. We benefit from the fact that Wikipedia is highly ranked by Google. And then in terms of contributions, currently we see an increase of about 10,000 words per month from over 1,000 edits, equivalent to roughly 230 full-length journal articles. So it's certainly choppy, but we like to believe that there's an upward trend there. So just to give you an idea of why, what could this be, right? Why are we so interested in the GeneWiki, right? I'll just highlight one page for a protein called relin. Okay? And most of this work actually was completely independent of our involvement. But just to highlight what eventually these pages could become, right? So relin, starts with this nice table of contents that covers everything that will be covered in the paper, just to give some more organization. It has images and diagrams here showing the expression of relin in the mouse brain. It has sections on, for example, the pathological roles of relin. Importantly, right, there are inline references to the literature. So for many statements, you will see a footnote that is one link away to a PubMed identifier. And also, you also have hyperlinks to related concepts. So if you don't know what Norman-Roberts syndrome is, right, you're in a larger ecosystem of encyclopedic topics. So, so just to underscore the point that this is a collaborative effort, right? This relin page was certainly now over 100 editors, hundreds of edits over many years. Other biomedically relevant articles, right, give you the idea that this is, truly a community effort. So I've gone pretty quickly through that, but what have we done, right? Let me just summarize what we've done so far, right? We've taken structured annotations and, generally speaking, structured data, and we've integrated that and reformatted that to put that into Wikipedia. We've invited free text contributions from the community, and what we wanted to next turn to was, can we complete this loop, right? Can we take the free text contribution and somehow work those back into structured contribution? Because structured annotations at the end of the day are the currency that informatics scientists really know and love, right? Pathway analyses or statistical analyses or so on and so forth. So, so to do this, right, we took actually a pretty naive text mining approach, and I'm just going to illustrate this by example, right? Showing a snippet from the web page from APLP2, and APLP2 starts with this sentence, which says APLP2 associates with antigen-presenting molecules like MHC class 1, yada yada yada, and enhances endocytosis. Okay, and that's what I've highlighted there, and that's a wiki link to the page on endocytosis. So this is what I would say is, we're strictly in unstructured world. But the APLP2, right, because we have a role in maintaining the structured data on that page, we know that that corresponds to this NCBI entree gene ID. Just by simple string matching, right, we know endocytosis corresponds to that gene ontology annotation, right? It's not crazy to think there's a candidate assertion there. At the time I created this slide, and I'm not sure, I haven't checked recently, but crazily, right, I mean, this annotation did not yet exist. And if you go in the literature, right, that inline annotation, you can go directly to the the paper that would support the creation of such an annotation, which clearly says that APLP2 increases endocytosis and stability and yada 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 of MHC class 1. Right? So this is one way in which we can complement biocuration efforts using crowdsourcing and community annotation. So using this very basic text-linked strategy, right, we were able to uncover over 6,000 novel gene ontology annotations, over 2,000 novel disease ontology annotations. These are all candidate annotations. So, you know, the next thing we turned to was to try to assess, okay, well, what is the accuracy, or what is the value of those annotations? Are they all garbage? Are they crap? Or do they actually have some value? And to assess this, we developed this evaluation strategy. And I'm going to go through, it's a little bit complicated, so I'm going to go through it slowly. The idea is that we would start with a go term, and we would get to a gene list, right? And so as an example, we would start with axon guidance. There are 264 human genes annotated with axon guidance. Using NCBI's link between gene and PubMed, we would go and find 811 related PubMed abstracts, and then we would forget how we got here. Okay, so let's just start now. Pretend we have these 811 abstracts. We would do our concept recognition strategy in this. We would do enrichment analysis. And the question is, in these 811 articles, would we see enrichment for terms for axon guidance, right? So for axon guidance, it turns out it works great. Of the 264 genes that are involved over here, 13 of them are linked through PubMed, very low false positive rate, and it leads to a very high p-value just by doing evaluating this 2x2 contingency table. Okay, so this is the general framework of how we evaluate Go annotations, right? Let me show you a system where it doesn't work as well, right? So 87 genes involved in muscle contraction, 251 PubMed articles from those 87 genes, but of those let's see, these 87 genes, right, actually none of them get linked through their linked PubMed abstract. Okay, so this is where we get a non-significant p-value. But here, if we bring in now the annotations we mined from the gene wiki, right? 87 gene articles corresponding to these 87 genes. Well, actually 6 of them do mention muscle contraction in the gene wiki articles, right? And if you combine the PubMed abstracts plus gene wiki, you get now a very significant p-value. Okay? So this is one example of where the gene wiki mined annotations improve enrichment analysis in this sort of gold standard type of analysis we've developed. Okay? So again, muscle contraction went from a p-value of 1 to 10 to the minus 9. I'm going to show that over here. Right? So we didn't want to just test this in one gene ontology term. We wanted to test this across many. But I'm going to plot the p-value, or PubMed plus GeneWiki, versus p-value of PubMed alone. So down here, it would be more significant if we're, are things that are more significant with PubMed plus GeneWiki. Here are more significant with PubMed alone. And we can see by adding in GeneWiki mined annotations, we systematically improve this sort of enrichment-based analysis. So it underscores the point that the GeneWiki mined annotations aren't perfect, certainly not, but they are of good enough quality to improve enrichment analysis.
So what you're saying, but the ones that appear above the line, though, if you use GeneWiki, it actually decreases
Slightly, correct.
Yeah. Okay.
But I mean, here, if we're talking about going from let's see, PubMed only, it's 10 to the minus 12 to 10 to the minus 10.
So is this any relationship to like how watching Fox News makes you dumber?
I don't know if I can comment on that. Again, I think at worst, right?
Yeah.
We aren't. I would say that the stuff above the line is stochastic variation, right? There's a little bit of fuzziness in there, but certainly down here, are things where it previously was not significant, right? So, for example, these were not significant at all in the PubMed only analysis, and moving off the line clearly shows how the GeneWiki improved. Okay, so So great. I hope I've convinced you up to this point that the long-tailed scientist can be a valuable source of gene annotation. But I want to now again come back to the perspective of an informatics scientist, right? Being able to use crowdsourcing, right? So everything I showed in that last analysis in text mining, we're doing something a little bit convoluted here, right? We are asking our community to take knowledge in the literature, put it into an unstructured form, i.e. free text, and then use text mining to try to get that knowledge out again, right? And that is akin to going from a steak to a hamburger and trying to reproduce the steak, right? In an ideal world, right, we would actually represent that knowledge directly without having to go through this text mining step. And so the crowdsourcing analogy to Wikipedia for structured data is actually a relatively new initiative called Wikidata. Wikidata, the stated mission is here to provide a database of the world's knowledge that anyone can edit. And essentially everything in Wikidata boils down to a structured assertion. Here saying that Berlin has a population of 3.5 million as of that particular date, according to this source, right? So it's essentially a database that is openly editable. Why is Wikidata notable, right? Well, you know, Wikidata, because it falls under this umbrella of the Wikimedia Foundation, right, understands how to scale to large data, right? So, of course, the Wikimedia Foundation, the primary thing that they do is Wikipedia. Wikipedia, right, is, I don't know, the 5th most visited site on the internet. I think they averaged out, it's something like 2,500 hits per second. They have millions of topics, right? They understand how to do things at scale. They have a variety of different related initiatives as well. But point being that for the future, right, Wikidata is one of the Wikimedia Foundation's foundational sort of technology things that they want to incorporate to support all of these other initiatives. Okay? So what can we say about Wikidata, right? I again showed you the very basics of the data model. This is essentially how they want to collect what they call statements, right? Every statement is has this particular structure. And currently, Wikidata has about 14 million items. So an item is a thing, right? You could imagine, you know, Berlin is a thing, San Diego is a thing, President Obama is an item in Wikidata, right? So this shows, just ignore the colors here for a second, but this just shows the growth of things since February of 2013. Again, a very new project out to January 2014. So we're currently at 14 million Wikidata items. It's hit this asymptotic slope. If we look at the total number of genes in entree gene, for example, there's about 13 million total genes there. Okay, so you know, we aren't orders of magnitude above what they already handle, right? So if we added, for example, one new item for every gene, I don't think they would complain too much. There are currently 27 million Wikidata statements, right? And this one is still growing quite substantially. As one metric of, well, how much biological knowledge could we interject into Wikidata, right? There are about 150,000 total gene ontology annotations. Maybe that's, you know, an order of magnitude off. Maybe we can go up to 1.5 million sort of biological statements that we might put into Wikidata. Still, you know, that would be totally fine within Wikidata, what they can handle. So let's dive into a little bit of what we could actually see within Wikidata in terms of molecular biology information, right? So here again is the Reland page for in Wikidata. Now this goes down much further, but I've just shown you the top here. And for those in the back, right, essentially what is represented through the web interface is essentially it says, you know, very structured statement. Reland is a protein. Reland is a glycoprotein. These are all objects, right? So you could find, give me all things that are proteins, give me all things that are glycoproteins, right? So these are database-like entries. Reland regulates neural development. Reland interacts with these other genes. And the important part is that all of these concepts, from Reland to these properties to these targets, can be linked to stable identifiers, right? So this is, you know, structured data. Of course, we as informatics scientists aren't interested in a web interface. We're interested in the API behind the interface, so you can query and get all these data back in a structured form. So this is sort of one avenue that we're pursuing in terms of the GeneWiki is can we get more biological data into Wikidata in a community and crowdsourced fashion. We have been participating in this molecular biology task force, if anybody's interested in going. But the real concrete thing that we tried to do in the context of a Google Summer of Code project this past year is to refactor the infrastructure that we already developed for the GeneWiki, right? We took these databases, we integrated them, and we reformatted them to get into Wikipedia, right? So the GSOC project was simply to do the same thing, but to get it into Wikidata. And so in the process of our GSOC project, we've only just scratched the surface here, so I'm going to be a little bit light on the data. But one key thing was to define what the Wikidata gene model was, right? And so what we settled on was essentially this. Every human gene would be an item, right? Would be an item, and every gene would have properties, from the chromosomal position to the symbol to the Entrez gene ID, OMIM ID, RefSeq ID, so on and so forth, right? And then we had relationships to the human protein, right? So a human gene encodes a human protein, and a human protein is encoded by a human gene. And this human gene would have properties, this human protein would have properties of its own as well. A uniprot ID, an EC number, ensemble and RefSeq protein IDs. And importantly, right, links out to gene ontology terms, right? These are hanging off of the human protein. And so this is a way we could have a structured database, community structured database for gene ontology terms. Because we want to represent the breadth of organismal diversity, we also added to that the corresponding mouse gene and mouse proteins through this orthology relationship, right? So this is the basic data model that we came into. There's a lot of work in terms of the infrastructure to put it together, and just discussions with the community about how to structure this. So far, we've added about 1,000 human genes, essentially with all these data that we got from the structured databases. So this is still an ongoing process. I don't want to oversell what we've done so far, but at least we haven't hit any insurmountable roadblocks. So let's tie it all back in, right? Can Wikidata be essentially that CMOD, right? Can we imagine putting all the structured data that we have available for not just the well-funded organisms, but essentially the long tail of organisms as well, and use Wikidata as sort of this, at least the database on which we can organize this genomic information. And to take it even one step further, right? You could even imagine CMOD eventually there being not only the single community instance for the database, but a single community instance for all the GMOD tools, right? So imagine for a second we had a JBrowse or a gbrowse instance, right? And there was a drop-down box that says organism, right? And in that, when you click that drop-down box, right, it goes and queries Wikidata. What sequence organisms are available in Wikidata? And you select whatever random organism out of the list of 3,000 or 10,000 organisms there. And once you select that, then it would query Wikidata and say, give me all the features and the genomic positions of all those features. And that would be the back end to a JBrowse instance. You would have the single JBrowse or gbrowse instance that could be, nobody would ever have to set up their own genome browser again, right? Wouldn't that be pretty cool? Again, going along the idea of GMOD as a service, as opposed to GMOD being simply open source software, which already is fantastic, right? Don't get
I wasn't offended.
Yeah. Okay. All right. So that is essentially what I wanted to the idea I wanted to try to communicate, right? The fundamental question is, can we harness the long tail of bioinformaticians to create a CMOD? And again, I'm very interested to hear input from the rest of the community here. These are all the people that were involved. I should particularly highlight Ben back there. I think Ben and I first dreamed up this idea in the airport on the way back from a biocuration conference, and it's been percolating ever since, and Ben's been very instrumental in the GeneWiki work overall, as well as sort of the ideas behind CMOD. These are the other group members and past group members who contributed to this project. Lots of people have collaborated with us on GeneWiki development. How to get in touch with us, thanks to NIH for funding and support. We do have possibilities for openings, so Scott and I should chat. But in any case, I mean, at many different levels, right? Students, postdocs, as well. So if you have people in your labs or colleagues who might be interested in sort of this interface between crowdsourcing and genomics, we would love the pointers. So with that, I'll happy to take any questions.
All right, let's go ahead, Ron.
What options are there for getting data out of Wikidata? That seems to be what really a lot of things. Can you do Sparkle queries against Wikidata? Does Wikidata produce good REF?
So, Wikidata has as its core principle, right, the idea of structured data. And clearly, because Wikipedia is incorporating Wikidata as a backend, there are certain parts of it that need to be highly performant, and I completely trust Wikidata to be able to make those queries highly performant, right? Because they're not going to tinker with they're not going to put Wikipedia on Wikidata until they're very sure it's going to work, and they've already started that integration. Okay, so having said that, it's still very early days, especially in terms of the query interface. To put it frankly, the query interface is almost non-existent. You can retrieve data for different objects, but to actually do the queries is a little bit more difficult. But this is something that they just haven't focused their attention on. Right? There's enough work to be done.
It isn't really part of their mission, really. They're making it seems like they're making a backend that's going to work for them. And it seems like they're not really focused on structured data that much.
Oh, so that I disagree with, right? They are very focused on structured data. I'm sorry?
The Wikimedia Foundation itself.
Well, this is their emphasis on structured data, right? This is their recognition that structured data is important. Now, it's a different question of whether or not they will be able to create infrastructure that can be live queried, okay? And that's something where I'm not sure. But certainly the exports to RDF, the integrations with semantic web, these are all the discussions that are happening within the community now. Now, I think the worst case scenario is that Wikidata is the aggregation interface for community annotation, and that gets exported into a triple store that has a Sparkle endpoint and becomes sort of the thing against which we develop. But ultimately, I think the first step is still a very important step, is how do you engage the community to crowdsource structured data? And I think this is a very viable source. I will also say that Freebase is another similar type of initiative that is now owned by Google. And they actually have a fantastic query interface. Well, fantastic might be overstated, but it's very performant, right? And it's another initiative, another infrastructure that we've thought about prototyping as well. Anyway, the pros and cons of Freebase versus Wikidata can be debated over beers. Yeah. Sorry, I skipped you.
This is part comment, part question. So I've been involved in many efforts to get users of databases and websites to contribute information inside and outside biology. Most recently, I worked for Maze GDB. The community is begging for annotations. It comes up in every meeting, and so the websites produce tools. My last job there, before funding ran out, was to produce another set of tools to get people to do annotations. Who uses them? But our curators who love them. There was an effort at one point to give gold stars to people who gave annotations, and we got some feedback saying we love this from the two people who are not part of the team who contribute. How did you get people to contribute?
So it's a great point, right? I mean, this is the most common comment that we get from skeptics, is that I tried community annotation, it didn't work,
I'm not
therefore
skeptical.
yours should.
It's gotta work, it's gotta work.
So I will say that people underestimate the difficulty in building community. And so there were several other GeneWiki related things that happened at the same time we were developing ours. The only difference is that we put our effort within Wikipedia, where the community already existed. Now there's trade-offs to that, right? We don't control that infrastructure at all, right? So there are different minuses to being within Wikipedia, but one thing that we didn't have to fight with was developing that critical mass. And that's a very challenging problem. I'll say also this, that it's not just the broad community of data generators who we're going after, right? So let's take an example, all the work that Mar just presented, right? You could, in terms of the similarity between different proteins and so on and so forth, right? That's all data that could be valuable to other people. And if other people wanted access to that data, right now, it's okay, I email you and say, can I please have a database dump? And then, you work on database format, yada, yada, and it's a big pain, right? Or, right, you and everybody else who's doing the analysis like you, just simply deposit your data into Wikidata, right? And then it's sort of a common infrastructure on which people can query. So point being, it's not just the data generators, it is the data analysts, right? The people in this room who take unstructured data and make it more structured or do computations, and they just need a place to put it so that now we can aggregate the efforts of all sorts of people.
I would just like to add a couple of things to your comments that are, I think, spot on. Other things that I've noticed, not necessarily with annotating genomes, but just getting people to write anything, like in a wiki, like in the GMOD wiki, is one, empty pages are anathema. That's why I think one of the things that the GeneWiki did that so brilliantly was creating a stub that already had something. That was the same thing we had with the GMOD.org wiki, is that it just sat there empty until we paid somebody to put stuff into it, and then other people put stuff into it. And the other thing that's interesting about using Wikipedia is that you have these people, and probably bots too, I don't know, but that maybe they don't know anything about biology, but they're formatting Nazis or grammar Nazis or something, and they will come in and clean stuff up so that you have extra editing power that you wouldn't get elsewhere.
Absolutely true.
Okay. I knew you'd agree. So, okay.
I'm just going to work from the front back as rewards to people who. Yeah.
All right. So my question is based on the fact that Scott is running into funding problems for Chado, and given how much GMOD supports a lot of things, that just seems ridiculous. But you can't count on funding sources not to be ridiculous. So with Wikidata, this is a weird sort of funding source with Wikimedia Foundation. So what does that mean? Because what their remit is not exactly our remit. And is the federal government going to be able to give grants to Wikimedia to support? I mean, that sounds like unlikely that NIH would give them a penny. Because hey, we have contracts.
For my speech later.
Yeah. So I mean, NIH likes doing a lot of things in house that are this scale. I mean, Entres, and there's good tools. So, I mean, that's my biggest concern, is that, we're dependent on this entity, the NIH, which does things both intramurally and extramurally, and, there's a giant in the room.
You raise a great point, and I don't have anything authoritative to say on it. I mean, I would say that, I don't think NCB, one of the things that NCBI doesn't do well, I think, is community stuff, right? I mean, they're great at data repositories, but they haven't.
Well, who needs the community when you are the authority?
Well,
The authority doesn't need a community, they're the authority.
Yeah, exactly.
but the argument then would be that NCBI should take advantage of all biocuration and do all Go curation, and they already have 100 mesh annotators, and that just barely scratches the surface of annotation of the literature, right?
Sure.
So I mean, I think your point is very well taken, right? And I would just say that, right, Wikipedia and the Wikimedia Foundation don't look like they're going anywhere, right? They seem as stable as anything else. And plus, one of the beauties of structured data, right, is that suppose Wikidata shuts down, next week. Well, we could get a data dump tomorrow, and it would suck. But we could figure out other infrastructure, or we could think about whether we wanted to find other infrastructure to support.
And I think part of the point you made somewhere there in the middle is that the amount of data we're talking about is small relative to what Wikimedia already deals with.
Right.
So they might not even notice. No, I don't. I don't think that's true. But I think that's a valid point.
Just briefly in relation about the last question, you might find you have less trouble getting grants from people outside the US, but it's not linked to things that are owned by the federal government. But
Yeah, you've
for
heard
people who
right.
aren't in the US, it's probably more advantageous to work with like MediaWiki because it isn't controlled by the US government. I'm not saying that's a bad thing, but some people are a bit funny about that. You were touching on data stored in papers. One of our groups, the Cosmic Group, actually, back in the days when it first started, they were curating straight for papers to try and retrieve all that information. And more recently, they've actually managed to engage with some journals, trying to get an enforcement that, especially if you're a small group who can't support high-tech hardware to provide FTP dumps and stuff, that your data has to go in to look at those mutations of cancer. It hasn't moved a lot. They've got 2 large European journals involved for genetics. They're working to get a framework put together for non-technical people to be able to submit data more easily. But they also provide all of the data from our database already as database dumps and Oracle XE instances, etc., etc. And it'd be interesting possibly to put you in touch with the developers because they'd probably be more than willing to try and inject their data into your project to look at that.
Please do. I think that'd be fantastic. And I think your point about publishers being involved in the eventual solution is a key one. Unfortunately, the publishing industry doesn't move.
They don't
very
have
quickly.
to enforce stuff, but
Yeah.
They're
if you
doing
can
it
get
more and
key
more.
journals,
They're doing it more and more.
And so there's a lot of analogies between what's going on with software reproducibility, and they're trying to get in front of or talk to publishers about, okay, we need to see code, we need to see things that people are actually using, know the versions of software that are being used, and get in front of it. And so if you do that with data, and say, okay, we want to have, for instance, the Operon file that you have available as a GFF or something that's easily integrated, and then that would be fantastic. There's so many pieces of data that just don't.
Yeah, and for those who aren't aware, right, Force 11 is actually a fantastic organization that's trying to work on this, the bigger issues of scholarly communication. But at the same time, right, these are things that we can actually do and actually control. And maybe they're stopgaps, but I think they will advance science sort of in that area.
One of the issues that I haven't heard you talk about is standardization. So the Go consortium currently is the gold standard for genomic annotation. And if you look at the Go annotation database, which is in Chato, I think, it's basically, it's not just gene and Go term and relationship. It's a bunch of other stuff as well. So there's actually more to a Go annotation than just what you're presenting there. So one of the complexities we found in our effort to do a community annotation forum is that people don't really grasp the extended nature of what they're trying to do. We force them to do it, and often what this results in is yet another spreadsheet submission, if there's any submission at all. So I guess my question would be, how do you, if there are community standards like the Go annotation standard, how do you get beyond the professional curation community in that case?
Yeah, it's a great point. So let me just first say on the data model side, right? Go annotations have evidence codes, have the PubMed IDs, have all the things. They're very important and actually underappreciated. I think those are all consistent with the Wikidata model. So one of the core things in the Wikidata are qualifiers, meaning qualifiers and the sources, right? And so I think all of that could fit in. But you're absolutely right in terms of community annotation, right? The more you ask of your community, the less participation you get. But this a little bit goes back to Scott's point, is that